doing to me? There we go. Okay, so where did we get to last time? I know we talked about this stuff, right? I think we were right around here.
Yes, the last one was
Okay, so we'll
this one.
start with that. That's a good place to start, actually. So let's go ahead and get started from there. Okay, so last time we kind of looked at the epidemiology of cancer. I mean, we talked about why it's so important to learn more about it. We talked about how it's the second leading cause of death in Western countries, and how, it is, what are all the different factors that cause cancer, that can lead to cancer. And there are a lot of them, right? So we talked about some environmental factors, we talked about some actual biological, bacterial, viral infections that could cause it. And we also talked about some physical carcinogens, actual, that can cause actual breaks of the DNA to cause the mutations like UV radiation and gamma radiation. So now we're going to talk about some pathways that are important for cancer development and progression. And then we are going to finish off by talking about a few hallmarks of cancer. So some main phenotypes and some main things that people look for when we are looking for cancer cell versus as compared to a normal cell. So we finished off last week or last class, last lecture, talking about how we get mutations all the time. However, they're constantly being repaired both by the DNA polymerase as it's replicating the DNA, but also by the DNA repair pathways as the different mutations are happening from these carcinogens that we are getting involved in, getting exposure to from environment. And those DNA repair pathways are obviously going to be very important to our body system's integrity. Um, Now, for cancer to occur, you have to have mutations in one of the five key pathways that we talked about at the end of class last time. And the first one and the foremost one is DNA repair. Now, one of the hallmarks of cancer is the genome instability, that the genome becomes haywire, right? You get one mutation that makes it more likely to get another mutation and another mutation, and it just ends up with all these duplications and translocations and just general mutations throughout your genome. Well, one of the easy ways for a cell to do that is if one of the DNA repair pathway is broken down because of a mutation. So if your primary mutation that happens because of the environmental exposure is in the DNA repair pathway, now one of your police is gone, right? One of the force is gone that is going to control certain types of mutations and repair them. Now it can do it. Well, that's all the more likely that you are going to get another mutation and another mutation. Another very important pathway for this, for cancer development, is obviously our cells' ability to regulate when they need to grow, when they need to divide, and when they need to be just hanging on, doing their work that they need to do, and when they're too old to live on and they can trigger their own death and just finish off. And those are in cell division and apoptosis. So anytime you have a mutation that dysregulates those regulatory pathways, whether they pertain to growth and division, as in cell cycle control, or well, the way they pertain to response to major damage through induction of apoptosis or programmed cell death, if you mess up those anywhere along those pathways, you are now more likely to have a cancer phenotype. And then finally, for more aggressive cancers, they have this uncanny ability to just go and survive in multiple types of environment and repopulate a different area. And that cannot happen unless they get two very important phenotypes. One is their ability to now survive as a suspension. So most cells in our body, they are hanging on to our matrix, right? They're hanging on to some scaffolding to keep them in place, and they cannot grow, just like cells in culture cannot grow in suspension, majority of them, right? White blood cells and some other cell types aside. Um, they cannot grow in suspension. They need to be attached to a substrate. Well, so one of the properties that cancer cells gain is that they are now able to survive at least partially in the absence of a substrate, and they can survive for some period of time just hanging on to, just floating in the bloodstream. And that allows them to travel and go to other organ systems, other places, and repopulate those spaces. And then finally, to repopulate another place or to go and colonize another place, they need to be able to survive in that new environment. And they do that by one of two ways. One is that some cells, when they're still in the earlier phases of cancer transformation, they may not have the ability to survive in multiple types of environments, but they could survive in similar environment organ systems. So they could go to those places. But other times, they actually de-differentiate. So they are more like an embryonic stem cell. And now they are able to go and populate multiple types of environments and just grow there. And so those are all important pathways that can be altered in a cancer phenotype. Now, just because you get a single mutation doesn't mean that you automatically get cancer. Actually, cancer cells evolve over a long period of time and through increasing competitive pressure put on them at a various level of their life cycle until they reach that cancer phenotype. So a single mutation rarely is enough to cause a cancer. Usually it takes 2 to 3 mutations minimum before you start to see transformation in a way that will lead to a cancer phenotype. And along the ways, there are many places where our body has check and balances to stop that from happening, to stop it where it is and either kill that cell off or to stall its growth so it may not produce more cells, right? However, that single first starting mutation does give that cell an advantage and gives it a little bit more chance to to gain that cancer phenotype down the road. So here is an example where the first mutation, all it's doing is maybe disrupting the cell cycle regulatory pathway so that this cell is now going to cycle a little bit faster than the normal epithelial cells around it. So those cells may only replicate every 36 hours or 48 hours or every week, while this cell is going to have that time shortened to maybe half the time or even a third of the time or a tenth of the time, but a different time frame, a quicker time frame than usual. Well, that higher turnover rate leads to a higher chance that they will get a second mutation because that's more copies getting made, more chances of mistakes, and because they're dividing in a shorter time frame, more chances of those mistakes not getting corrected in time. Well, because of that, it obviously gives it a second mutation. Now, that second mutation may be innocuous, may not be something important, in which case nothing happens. But that second mutation may be in another one of those key pathways. Maybe this time it was in a pathway related to, instead of cell cycle, it was related to one of the other phenotypes like apoptosis. So that inhibiting, it's going to not be able to complete apoptosis all the way, maybe. And that second mutation now obviously provides it with a secondary advantage, right? Or it may be even more dangerous mutation like a DNA repair pathway. So not only is the cell cycling faster than usual, but now it also have a mutation in one of the key DNA repair pathways. And now it won't be able to fix other mutations as well. So that in turn would then make it even higher chance for the cell to survive and proliferate at a higher rate, accumulate mutations at a far higher rate. And usually that third mutation is what will then make it a aggressive cancer or make it able to invade other cells. So we see an example of this all the time in any type of cancer that we look at. And we can see the importance of that first starting mutation, and the timing of that first starting mutation when we compare hereditary cancers, so cancers that run in families, versus the same type of cancer that just starts in situ without any hereditary component to it. So typically, if you look at colon cancer development, it usually takes 2 to 3 mutations before you see a cancerous adenoma. Usually, there's a first mutation is in the APC gene, which relates to the regulation of cell cycle. It makes that the epithelium of a normal colon proliferate a little bit faster, gives rise to those polyps and benign adenomas, as we would call it. So they are benign, they are not cancerous, but they are extra growths. The cells are growing and accumulating at a higher rate. Well, usually that leads to a second mutation way down the road. So once an APC mutation happens, it usually takes anywhere from 7 years to even 20 years before that second mutation occurs. That second mutation, we've noticed many times, is one of the key cell cycle regulatory genes, like KRAS, which, if you remember from last week when we talked about cell signaling, is part of that growth response pathway, where it is a proliferate, it's a mitogenic pathway. So this is going to induce higher growth rate, leading to a nodule that may be that early cancerous nodule. And then over time, you have a secondary loss of a DCC gene, which makes these cells a little bit less, needing the substrate. They can slough off very easily. They can now move. They can go other places, and lead to a more cancerous phenotype. So if you are born, if you in a family with hereditary cancer history for colon cancer, you are usually born with the APC mutation already in place. So because you're born with this mutation there, it kind of accelerates your process. The time from the first to the second mutation, while it's variable, but you know that once this mutation is there, that second mutation will happen eventually, right? But this is usually the longest time that you have. After that, once KRAS mutation is on, the other mutations happen pretty quickly afterwards. So by having this mutation already in place at the time of birth, you essentially know that your chance of getting that secondary mutation at a young age, in 20s or 30s, is a lot higher than a family that wasn't born, than a person wasn't born with a familial history for a colon cancer, who first has to get that first mutation in order for this process to start. Does that make sense?
Yes.
Okay. So now, just going back to the hallmarks of cancer and making sure we understand those 5 main hallmarks of cancer. In the article that you will read will talk about secondary hallmarks of cancer as well, but these are the original, the basic hallmarks that all cancer cells will show. The first one and foremost is self-maintained replication. They do not need external growth factors in order to replicate and divide. They have a mutation in some key pathways related to cell cycle regulation, so that now they either have a lack of response to inhibitory factors, or they have one of the receptors mutated in a way that it's always switched on. So it's always sending, like the RAS gene, always sending a growth signal to the cell to grow and replicate and divide. Another one is longer survival. And that simply means that they have somehow bypassed the main programmed cell death, the apoptosis pathway, either through a mutation in the apoptotic pathway, in one of the apoptotic pathways, or on the other hand, they can also do a secondary way, which is the telomerase reactivation, which, as we know, is not active in normal living human beings once they are born. However, in this case, they reactivate telomerase through a mutation in that gene, so that now it's always active. It's always adding those telomeres at the end with each replication cycle. So the cells have this false sense of youth, right? They think they're still young, even though there's mutations, they're still damaged, all that stuff is still happening. But because the cells still have these telomeres, it falsely triggers the cell to still act as a young cell and replicate and divide like normal. Now, the third thing is obviously, as we talked about with defective DNA repair systems, if you have a mutation in one of the key repair systems that we'll talk about today, that obviously leads to genetic instability, which is the main problem with these cancer cells. That one mutation is now making them more prone to the second mutation. That second mutation is making them more prone to the third mutation. And with each one of these mutations, they are more and more able to survive in different environments and do more aggressive things as a cancer cell. And then finally, you have the last two, which are capable of inducing neoangiogenesis and invasion and metastasis. These two are specific to cancer cells that are now able to metastasize, not just grow where they are, but are able to come off, go into the bloodstream, go to some other place and replicate and divide there. To do that, there are two main things they need to do. First of all, in order for a cancer cell to grow beyond a very small, like 1 centimeter, diameter size, they need to be able to have enough nutrition and oxygen provided to them. Our own body's blood vessels cannot do that, right? They are meant for our body system. We didn't make roadways for a random building that is now coming in the middle of nowhere, right? You have to make a road to it. And that road is provided by the cancer cells secreting signals, right? It's like us sending letters to the government saying, hey, we have a new village here, we need some roads. Same thing here. The cells themselves, the cancer cells, send signals in the form of secreted growth factors that signal the blood vessels nearby that more blood vessels need to grow towards that gradient. And that's their ability to form new blood vessels, new angiogenesis, blood vessel growth. Um, and that now allows them not only to get the nutrition and oxygen they need to grow, but also, guess what? Now they have a road to go from one place to another. And they can go wherever they want or wherever they can survive inside the body. And finally, with this newfound ability, they can invade and metastasize. But invasion and metastasis doesn't just mean they have to have the roadway to go somewhere. They also need to have the ability to chew through the matrix, to chew through the blood vessel, to kind of penetrate through it, to survive in that bloodstream as a floating cell rather than attached to a surface. And all those things are taken through dedifferentiation and through other capabilities through loss of cell-cell contact and all that stuff that allows them to survive and do all these extra capabilities to metastasize and invade other systems. I think there's a question in the chat. Is there? I couldn't see it, unfortunately.
Oh, I was just...
Yes, the cells, this is exactly when the cells would invade the lymph system as well. And actually, lymphatic system is a simpler system to invade. So that's where they usually start. And then later on, they end up going into your bloodstream as well as they become even more dedifferentiated and more capable of doing that. Okay.
Interesting. Thank you.
No problem. Yeah, so cancer cells, basically two main things that we need to remember from them is that one will proliferate excessively, die less, and migrate inappropriately, right? They migrate to places they aren't supposed to be in. So here you can see a normal epithelial cell. It's growing in its place in an organized fashion, while when the same epithelial cell becomes mutated, it starts to pile cells on top of each other unnecessarily. It's able to break through that basal lamina, through the connective tissue, chew the matrix away in order to get into the bloodstream, where it can then cause metastatic tumors in other parts of your body. So here you can see the difference between a normal liver tissue versus these large nodules that are forming that are metastatic tumors from some other place. These are not liver cells. You would look at these cells, and for example, if they were coming from, let's say, breast, these will look like breast cells. They'll still have some remnants of that morphology, So you can identify where the primary tumor was coming from based on what they look like right here in the metastatic tumor. And sometimes that's all you find. That's what you find to begin with, is the metastatic tumor and not the primary tumor. Um, Sometimes, especially for breast cancer patients that haven't had their mammograms, that haven't been taking care of themselves and doing their own checks, they will sometimes find these secondary tumors first. The patient will go because they have a tummy ache and the liver is enlarged, and they check the liver and there's some tumors in the liver. They do a biopsy and they have breast cells in there, and then they go back and they're like, there's breast cells in there, there's a breast tumor somewhere. So that has happened more than one time. Same thing has happened with melanoma that have gotten undetected. Any type of tumor, really. There are many times that you find the secondary tumor first, the metastatic tumor first, unfortunately, because that has invaded a system that is more essential, or is causing more symptoms before they find the primary phenotype or the primary tumor, primary mass. So again, here you can see the difference between a normal cell versus a cancerous tissue, right? So when you look at the normal liver tissue, it's highly organized. The cells all look very similar to each other. They are not going to, they're going to be within a range of size and shape, and the nuclei will be all nice and clean and round. But then when you get into the cancerous tissue, you see not only that the cancer cells are much more, misshapen, the nuclei are misshapen, the nucleoli are messed up, but also that you have these large areas of kind of necrosis, these large empty spaces. These are actually necrosis that have happened in there. That's the cells were dying because they weren't getting enough oxygen. There was some other damage to them. So you will have really misshapen areas in these tumor masses. They're not all cancer cells. So now going to those mutations that are causing these cancers, somehow my chat is not showing. Yes, there are several things that determine where a secondary tumor will form. One of the first places that the tumors tend to go is downstream and right the next organ downstream and upstream blood system wise, right? And that's for clear reasons. As the blood is flowing through, it carries them to the next space and they get there. If they can survive in that environment, that's the best place they'll grow. Secondly, they can go into similar environments. So breast cells love to go to brain, love to go to lungs, again, because of proximity, next upstream, downstream organ system. But they will also go into similar other systems as well with similar environments. Your cervical cancer will, and ovarian cancer, they'll all travel to breast much easily. Digestive system, if you get it in a stomach, it's going to be much more likely to go into other spaces like your esophagus, like your intestines, and go from there. So you'll see them going into the next primary organs in similar environments. Bones are usually one of those organ systems that they go into at the end, and mostly all of the cancers can eventually, if they differentiate enough, they will end up causing masses in the bones. And obviously those are the hardest one to treat as well at that point, unfortunately. But that's usually a sign of a very, very aggressive, very late stage tumor if they've gone to the bones. Okay, did that answer the question? Okay, so two main classes of our, mutations that we see in cancer. One is a class called oncogenes. Oncogenes are essentially genes where if they are mutated, they will lead to a cancer phenotype because of their own typical normal function being upregulated. So think of a growth factor or a growth factor receptor that's usually regulated in a very strict way regarding when it's going to be active and when it will give a mitogenic signal. Now, its role is to give a mitogenic signal. Well, if it gets mutated in a way that now it's always active and that activity leads to a higher growth rate, that's an oncogene, right? So that's an example of an oncogene. Now, in this case, remember, for every gene, you have two copies, one from your mom, one from your dad. If even one of those get mutated, you're in trouble, right? Because even if it's one is sending that signal all the time, that's enough. It's sending that signal all the time to your cell. On the other side are your tumor suppressor genes. Now, these are regulatory genes. These are the genes that are regulating those oncogenes or other functions that are important for your survival. Now, the tumor suppressor genes, their normal role is to stop a mutation from happening or to fix it or to regulate it in some way. In this case, if you get a mutation, again, you have the two copies. If you get one of the copies mutated, you still have the second copy doing its job. You still have somebody keeping check, stopping it. There's only one guard there instead of two, but there's a guard there. However, if both copies get mutated, then you are in trouble. So here is an example when, if in a familial cancer, you have one copy mutated at the start of your lifetime, then you're more likely to get a second mutation in that remaining copy. When you have that second mutation, then you will have a loss of function mutation. So oncogenes are generally dominant mutations. They are gain of function mutations. So as soon as a mutation occurs that always makes this active, it's going to lead to excessive survival, proliferation, or both. In recessive mutation, a tumor suppressor, on the other hand, is a recessive mutation. You need to have a complete loss of function for it to show as a cancer phenotype. In this case, if one copy is mutated, you'll largely have no effect or very little effect because there's still a copy keeping check. However, if both copies are mutated, then you will have trouble. That's when you will see the tumor suppressor gene not working properly. So how we make an oncogene could be multiple ways. You can have a simple mutation in the coding sequence of one single amino acid sequence, amino acid getting mutated in the key, catalytic site of that protein or in the active site of that protein. And you can lead to a protein that is hyperactive mutant that is always active, always made. It's made in the normal amount. It's made just like a normal protein, but it's always active. It's not regulated anymore. You could also have a event called gene amplification, where that gene is duplicated or triplicated in the genome due to mutation, due to a slippage of the polymerase. And you can lead to multiple, way more protein getting produced. So you have a normal protein, but it is overproduced. So when it's activated, it's overactive because of that. That's another way this can happen. And finally, you can have an event called chromosome rearrangement, where you can either um, have a mutation, a translocation in a way that the nearby regulatory sequences are now always keeping it on or always activating it in a way that it's always active or produced a lot more, leading to the phenotype that you see. Now, in a tumor suppressor, it's a different way, obviously, because here you have to mutate both copies. One of the ways, the easiest ways that you see it happening is because you are born with one of these mutations already in place. Maybe you are born with one of the mutations there, and then when the second mutation happens in the second copy, you have a complete loss of that tumor suppressor. Another one would be that maybe the whole chromosome gets lost. Maybe it's a gene on your X chromosome, and in a male, you may see that it is getting, if it's mutated, they just needed one copy mutated. That's all the X they had, and it shows up a lot quicker. Or in females, maybe the Barr body happens in a way that the mutated copy is the one that's active in a cell, and that leads to obviously a bigger chance for that one to end up with a cancer phenotype. You can also have a normal gene deleted from your paternal chromosome, right? This gene could just get deleted out because of a translocation event or a mutation event. Um, or you can have, like I said, mutation in both genes. Finally, you can also have a situation where it's perfectly fine. However, those methylations we talked about, right? You can have a hypermethylation effect. It's a epigenetic mutation where the methylation has improperly inactivated it and silenced it, so that now you have a complete loss of function, leading to the tumor suppressor phenotype. So here is an example of how a tumor suppressor like APC, like P53, getting mutated can lead to a very cancerous phenotype in the colorectal, right? And all these polyps, many of them are going to be cancerous over time, but some of them are cancerous right there. And then when you look at that epithelium, you see how disarranged they are. And it is not the typical normal epithelium of the colorectal site. So looking at this, how it would have happened. Again, we are looking at the APC protein, which is one of the first proteins to get mutated within that pathway in many of your colorectal cancers. Now, there is another protein that it regulates. The APC protein's job, it's a regulatory protein, is to regulate multiple mitogenic pathways. One of those is Wnt signaling pathway. So we talked about EQT pathway before. Now this is Wnt signaling pathway. So the Wnt signaling pathway is kept in check by the APC protein and kept inactivated unless needed, so that you don't have a lot of overproduction of the epithelial cells in that region. Now, when you don't have Wnt signal, it's the Wnt receptor is inactive, and that is that inactive signaling protein is not going to obviously is going to bind to the APC containing complex, right? And that complex leads to binding to other proteins like beta-catenin and target them from degradation, so that they don't activate downstream gene expression. However, when there is a protein bound to the Wnt signaling pathway to activate the Wnt receptor, the active signaling protein can no longer interact with APC, right? So the APC complex is going to be inactive in this case, and you'll release the beta-catenin. It won't be able to bind to the beta-catenin because it needed the inactive signaling protein to bind to it. And that will release the beta-catenin into the nucleus, where it can regulate gene expression. And that leads to proliferation of the gut epithelium. Now, if you have, for example, some kind of an infection, inflammation in your colorectal epithelial cells, this signaling pathway is active at that time to signal to the cells that there is some damage that needs to be replaced, that needs to have proliferation. But outside of that, your APC protein is keeping it in check so that it's not activated all the time. Okay, so here are some critical examples of cancer, most commonly mutated cancer genes. You should know the names of these genes, and you should know whether they are oncogenes or tumor suppressor genes. Proto-oncogene simply means that, it's a precursor to the oncogene. Normally it functions in a regulated fashion until something goes wrong. So RAS, beta-catenin, those two are your main oncogenes that we're going to be repeatedly talking about the rest of the semester. And then P53, APC, and BRCA1 and 2 are tumor suppressor genes that we will talk about again and again and again. P53 gene is actually the most commonly mutated, either direct or indirect, common pathway in all cancers, regardless of their origin. It's directly mutated in over 60% of the genes of the cancers, and it's indirectly and directly combined mutated in almost all cancers. So that is a very, very, very important pathway, and we'll learn a lot more about it throughout the semester as we talk about different pathways and how they are regulated in those 5 critical signaling cascades that we talked about related to cancer. Okay. Now, many of your current treatment options rely on that p53 activity in order to be active. So usually if you cannot surgically remove your tumor, either through minimal invasive surgery or through invasive surgery, your best option next is either chemotherapy or radiation. Both of those rely on p53 pathways, at least some of them being active, in order for best efficacy. Yeah, otherwise, there are a lot of new techniques that are coming out, new treatment options that are coming out that involve either stem cell transplantation after regulating them and creating them a certain way, immunotherapy, where we train our own immune cells to target the cancer cells and kill them, as well as targeted therapies against certain mutations that are present inside the tumors, so that only cells that have that mutation are killed off and your normal cells are kept alive. Um, so that's something that, again, we will talk about again and again throughout it. So these are some of your emerging therapies that are finding. Because at the end of the day, when we look at our cancer cells, no two people will have the exact same mutations. No two people will have the exact same therapy that is okay for them. Different mutations lead to different resistance in these cancer cells. And at the end of the day, if you think of a mass of cancer, that contains multiple cells with multiple mutations. You have some cells that have only one mutation, some cells that may have 2 mutations, some cells that have 3 or 4 mutations. And some of those cells, a subtype may be sensitive to a drug, but none of the drug, not, not none of the drugs are going to be sensitive, are going to be effective for all 100% of the cells in that tumor mass. So that's why many times when you look at cancer therapy, one, people are looking for more and more targeted therapies to these exact mutations, but also many times you will see multiple drugs being used as a cocktail in cancer therapy and not a single drug as the master drug, because there's no single drug that can act on all these mutations, right? So this is just telling you how conventional therapy, which is just the surgery, chemo, radiation, is a little bit more general approach, but that's only applicable if you have a really tiny tumor that can easily be removed with all the margins clear and can get minimal chemo and radiation. Otherwise, it's going to cause a lot of toxic damage to your body. However, neotherapies are looking for those targets that they can target. It's biomarkers, actual protein targets they can target, so that they can give them these in addition to these conventional therapies and have a better chance of survival for the patients. Questions on this part?
Why sometimes they say, like, if you do a surgery, uh, then the cancer cells will proliferate to different parts of the body?
So what happens is that if you have a really large mass in place 1 or place 0, the starting site, that big mass is requiring a lot of energy from your body in order to stay there and grow and continue to replicate and divide. And so that takes away energy from all those circulating cells, the cancer cells that are already circulating in the bloodstream, and they have less ability to grow and divide. Sometimes when you take that large mass away, those few pockets of cells that were just hanging on, but not surviving, but not growing, now have that influx of energy provided to them, and they can grow like wildfire. So that's why if the cancer is at a size that it is not going to be easily operable, they prefer to first treat it with chemo radiation to reduce the size, get it manageable, target those other cells that are circulating as well, so that because they're already at a weakened state, if you can kill them off, you have a better chance that they will not return. But if you take away the mass and haven't started treating them first, those cells, they will grow, unfortunately.
Okay, got it.
Okay. So yeah, so our molecularly targeted therapies, these are the things that they are focusing on. Is in the cancer cell, they're either going to inhibit angiogenesis, so the cells don't get new blood growth and they cannot get new oxygen and more nutrients. Obviously, they somehow try to inhibit their ability to invade and metastasize. Some of them actually bulk up your extracellular matrix, so it's harder to chew through. And then some of them will work with your immune system to provide it with that immune response that our own body can launch against these cancer cells in order to target them and kill them off. Okay. Okay. So that was now your cancer biology intro. And we are ready to go on to our what is it called? Our DNA repair. Would targeted therapy require knowing what the mutation is? Yes. So what we do is, so there are a lot of targeted therapies in the market nowadays where we know what type of mutations they are effective against and what type of mutations they can actually have the opposite effect. They can actually help them grow more. So the first thing usually that happens once a tumor is identified is they usually run a biopsy and then run genetic analysis on it to identify all the mutations that they can find within that set. When they look at those, they do that through immunohistochemistry, they do that through genetic testing. They then meet, actually, anytime someone is diagnosed with cancer, they have a whole, and any organization, they'll have a cancer panel. They will discuss the patients as a whole. A bunch of, so they have immunotherapy doctors, they have immunologists, they'll have the radiologists, they'll have their typical surgical oncologists. They'll also have palliative care and other folks already in that panel. They discuss all this patients together. They look at all their mutations and come up with the best targeted therapy for them, or the best regime, therapy regime for them, based on the type of mutations and the aggressivity of the tumor and where the starting point is. Okay.
Is this kind of the most I'm sorry.
Oh no, you're good.
Go ahead.
See you guys. I was trying to see you guys.
So I have a question. It's kind of like confusing. I don't really know how to word it. But so like when you get like the Oncotype or the Oncogene test for like breast cancer, and it tests for like the BRCA1 or BRCA2.
Mm-hmm.
So it's not like the BRCA1 and BRCA2 since they're considered like the tumor suppressor genes, so they're not actually the oncogenes.
So BRCA1 and BRCA2 are tumor suppressors. Mm-hmm. But again, like if you're born with familial history, you're born with one copy already mutated, right? Mm-hmm. And so you have a lot higher chance of getting that second copy mutated and resulting in a cancerous phenotype.
Okay.
My question was about the target cell therapy. Is it like too much expensive because it's specific for each patient?
It's, well, so targeted therapies, we're going to start sharing some of those ads and things with you guys to look through and see some of these targeted therapies. I actually put them on the exam as well. So you'll see them again and again. But there are therapies on the market that, for example, will work for HER2 negative, PR positive tumors, right? And so once you know that somebody has that, that's the drug you want to give them. And then there are other therapies that work with your immune system. So in that case, they actually take your blood and they take your immune cells, they train them outside in cell culture to the cancer cells by giving them pieces of the cancer cells as epitopes to build antibodies to them. And then they inject them back into once they train them, they inject them back into your body where your body can then launch a response against the cancer cells. And that's something super cool that they are able to do. And in some cases works very effectively and with a lot less, you'll have your typical response as you do with the immune system. You'll have your fevers and chills and all that stuff. You'll feel like you're down with a bad flu, but you won't have the same type of damage done to your normal cells that you get from chemo and radiation.
Exactly.
Yeah. My cousin had melanoma and was treated as part of her regime with immunotherapy. And this was back in her 20s. She's only a couple of years younger than me. So that's like 15, 17 years ago. And she's been good since then. And when it was found at a grade 3. So it had already started to move, but they were able to treat it because they were able to use, at that time, this had just started, and she happened to work in a dermatologist office. So they were able to provide it to her on one of the clinical trials. But that is something that does work very effectively for a subpopulation of cancers. Okay.
Okay.
Everyone good? Yes, some treatments work really good wonders. Unfortunately, there are certain tumors that are hard to treat that still that still have a way to go before they will be treatable or manageable. But there are other cancers that are, even at a metastatic level, very manageable for several, and do increase life expectancy and quality of life for several years, even a decade, 15 years. Okay. Okay, so moving on to our next topic. I should increase my share again. Share screen. Okay, so now we are going to talk about one of these important pathways. So one of the things that we're going to do in this set of module, this first lecture that we talked, where we talked about cancer, is kind of our overview of this set of module lectures. We are going to be spending the rest of our 4 or 5 lectures talking about these different critical pathways and how they are normally regulated, and then what happens when cancer occurs. And so this is going to help you understand how important it is to keep that regulation. And hopefully, because we're talking about what happens when they're dysregulated, it will help you see step A to B to C connection as well. At least that's my thinking about it. So what we are working on this week is DNA replication and repair from chapter 6. And then our additional topic, so-called, that we have in here is how cells respond to damage, which is not something that's part of 6 on its own. So let's remind ourselves about the DNA replication machinery. There is a little animation of that as well that you can access through that link. So remember that when our DNA is getting replicated during the S phase, it's going to have, the first thing that will happen is it will unwind the DNA. And then there are many proteins that are working together to keep that DNA opened up. The single-stranded binding proteins are hanging on there. You have the helicase that is unwinding the DNA. You have the topoisomerase that is causing actual breaks in the DNA to release the tension in the double strand upstream and downstream of that helicase where it's working. You have the DNA polymerase obviously copying the DNA and proofreading it. And then there are other proteins that are working along with them as well, right? So here you see your leading strand where the DNA polymerase is just going in a single line. And on the other side, you have your Okazaki fragments that are happening with multiple RNA primers being settled in by the RNA primase, as opposed to the single primer placed up here. And so that is something that is going to be happening, right? So a lot of different proteins are taking part in this process. It's not a single man's job. So there are chances of happening mistakes, right? Mistakes happening because there's so much involved in just the simple process of replication. And there is so much DNA. There are billions of nucleotides to be replicated in every single cycle. Now, mistakes need to be fixed, obviously, because while much of your genome is not coding genome or doesn't code for an actual functional protein, anytime you get a mutation in one of those coding regions can have very, very, very real effects. So for example, sickle cell anemia is due to a single amino acid changing from, single nucleotide change mutation in the beta globin gene. A single mutation is enough to cause the blood cells to be completely misshapen and unable to carry enough oxygen and enough hemoglobin to the rest of your body, and have a host of other issues along with it, right? Sorry. Um, Same thing is with cancer, that a single mutation can lead to a very, very real cancerous phenotype. Now, there are many different types of DNA damage that you can get. You can get DNA damage due to endogenous factors, right? Endogenous is where it occurs inside the cell without any outside influence. Forget the environment, it's all because of what's happening inside. The biggest reason, replication errors. All that machinery working together, a particular nucleotide gets missed as it's copied incorrectly by the DNA polymerase, and that adds that incorrect base, which leads to mutation down the road. You can also have spontaneous base changes to the base structure. Certain base changes can happen quite normally. They can be fixed quite normally as well, but sometimes they get missed at some point, and that can lead to an issue as well. And then finally, strand breaks, whether they are due to oxidative damage from the reactive oxygen species inside the cell, or because of the topoisomerase nicking the DNA and then never somehow missing that fix. It could be because of those breaks that lead to duplication or translocations or mutations. And then other ways is through exogenous manners. Now, the exogenous influences are going to be pretty much the same as what we heard about in the cancer biology one, right? What causes cancer is what causes these exogenous DNA damage. And these are through external or environmental sources. They can be chemicals that alter actual structure of the DNA bases, or there could be radiation causing, breaks in the DNA, single or double-stranded breaks, as you see with the UV and the ionizing radiation, right? So it is important for us to recognize those reasons to know exactly what type of damage might have occurred, so we know which pathway is going to be activated in response. Now, DNA is getting damaged constantly in our cells, right? Even when they're just senescent cells, they are not replicating, they are not doing anything major, they're just hanging out, doing their work they need to do. You'll constantly have some type of a turnover type thing happening, right? So, two common things that can happen inside your bases or inside the DNA is what we call depurination or deamination. So this is when you are just causing the your purines to kind of come off the DNA strand momentarily and then be having to put back in, or deamination, where again you can have the amine group of your purines and pyrimidines popping off and on. So these are two common things that can happen just because they are living in an aqueous environment, and the water can react to oxygen and amine groups in these bases, and that causes issues with that. So cytosine can, through deamination, change to uracil. And now imagine a situation where you have this happen, and that's just in time for DNA replication. This one is going to put a, what is it going to put against it instead of a G? So if it was supposed to be C and now it is a U, what will the DNA polymerase put against it when it replicates? Put it in the chat, and I can see the chat. I'm sorry, I'm having A, exactly. So yes, you're going to end up with an A instead of a G, which is what you would have gotten if it hadn't happened. So that's a big change. That's a nucleotide that is different, and it can lead to further downstream issues. So the other thing that can happen is what we call thymine dimers. If there are two thymines adjacent to each other in the DNA strand, just UV light exposure from sunlight can cause what we call thymine dimers through a bonding between the carbons in the actual thymine itself. And that can obviously lead to over time, well, that complex, if it's not fixed, is going to lead to mutation when it replicates. So here are again how this will show up in real life. So this is a schematic to show you that when you have chemical modifications of nucleotides and they are left unrepaired, obviously that's going to lead to mutations. But that the biggest reason for that is a nucleotide shift. However, here you can see that if you have a deamination change from a C to a U, now the base pairs are not going to be able to base pair properly, right? Because you cannot base pair effectively with G. And what you will end up with is a bulge in the double helix. Well, that should be recognized so that it is fixed. And if it is not recognized and fixed in time, then in DNA replication, one of your strand is going to give you completely normal sequence. One of the cell will have no mutation. However, the other strand where you had the U will end up putting an A across from it and lead to a mutation, a single base pair mutation inside this sequence. Another thing that can happen is the depurination, where the purine is completely getting removed. And again, here in this example, you have A that is completely removed from that sequence. Well, again, if that is not repaired in time, one of the sequence will remain unchanged, but the second sequence, the second cell will get a deletion at that AT set because there's nothing against it. It's just going to skip it and keep going. And that can cause a major shift in the protein that it was going to make if it codes for an actual protein, because all the codons downstream of it are going to be affected by it. In this case, in the first case, only a single codon may have been affected. But here, every codon is going to be affected going downstream from it. So errors that are made during DNA replication itself are going to be similar in some ways, right? So when DNA is getting replicated, it can accidentally put the incorrect base pairing. Again, usually that is recognized by the DNA polymerase proofreading because it causes bulging, so to speak, of the strand because they don't base pair properly and causes it to stall and recognize that there's a mistake that needs to be fixed. However, if the replication is happening without repair, that strand now is going to become a permanent mutation in the second round as the bulge is fixed with the correct base pair against that incorrectly placed base pair. So here it went from a CG to an AT because it took away the G and replaced it with a T, leading to, again, a nucleotide mutation. So we can see actually a record of how good DNA polymerase is in repairing these damages that are happening, the mutations that are happening, which we can call the fidelity of the polymerase or the replication process, by comparing genomes of the same gene over time in various species. And we can see that majority of the genome is very, when you look at typical genes, they are very well conserved. And that shows how good DNA polymerase is at finding and fixing these mistakes. But like I said, there are times where you will see this type of mutation happening. And you can recognize it because of their usual, typical changes that happen, C to T, A against G. And that lets you know that somewhere along the way, there was a mutation like this that went undetected and led to the sequence that we have in our organism today. Now, how do cells fix damage? Well, there are actually multiple ways for cells to fix damage. There's no single DNA repair pathway. There are multiple DNA repair pathways, each one against a very specific type of mutation. How are we doing on time, by the way? Okay, so we have about 5 minutes, so we'll get to a stopping point and then continue next time. So for each type of damage that can occur from the DNA damaging agent, there are specific DNA repair mechanisms. So this kind of gives you an overview of all those things that we're going to be talking about in this one. So you have DNA replication due to stress that can cause just, base pair mismatches, or, you can have oxygen radicals, ionizing radiation, or chemotherapeutics that can cause a break in your genome. You can have, aromatic hydrocarbons and UV light that can cause these thymine dimers and other DNA adducts where they kind of, combine together and get cross-linked to each other. And then you can have ionizing radiation and chemotherapeutics that actually cause a double strand break, not just a single strand break. For each one of these, base mismatches, single strand DNA breaks, DNA adducts, and double stranded DNA, there's a different pathway that repairs that type of damage. So for base mismatches like insertions and deletions, you have mismatch repair pathway. For single stranded breaks, you have base excision pathway, where that improper base is excised and repaired. For DNA adducts, where you have those crosslinks, you have what we call nucleotide excision pathway, where the entire nucleotides is removed. And then for double-stranded break or interstrand crosslinks, you have double-strand break repair, which can happen through homologous or non-homologous ways. And we'll talk more about those later on. So this is the first one is just the basic mechanisms of DNA repair and the 3 steps that they are going to involve, regardless of what way it's going to fix. The basic, once the problem is recognized, the basic pathways will have 3 steps. The first is excising the damage, taking away the damage, whether it's just the base or the whole nucleotide or that entire portion of that DNA. The second is resynthesis, filling up that gap by repairing it with the DNA polymerase, filling in the missing nucleotides using the second strand as the template. And then ligation, where it seals the nicks in that genome and in the DNA strand so that it's a single strand instead of a broken space in between. Now, we'll start by looking at the simplest of these repairs, which is the DNA mismatch repair. Here, you had one strand, the original parent strand, that was completely fine, that was replicated correctly. And the second one, where you had a mutation happen and an incorrect base pairing was done. The new strand with error, for example, if you have the A to G, is going to end up with that improper base pairing will cause a bulge in that DNA strand so that it causes it to halt. In this case, the mismatch repair will only take out the nucleotide, the base that is mismatched, and fix the original sequence, restore it by putting in the correct base pairing, right? So in mismatch repair, you have enzymes that remove the portion of the DNA containing the mistake, and they follow it up with gap DNA polymerase filling up the gap, and then ligase filling up the links. So the mismatch repair proteins are going to basically cause a nick in downstream portion from the DNA. So they don't just repair one little base pair, right? They kind of come together and they remove that whole little area. They loop out from the nick to the base pair mismatch. They take away all of that sequence, the newly synthesized sequence, and then they repair it using DNA polymerase and ligase, just like they would in a normal situation, right? So again, it's going to fill it up and then you will fix it. Now, because in this case, you have DNA on both sides, you can just fill it up without the requirement of a primase putting in a primer, right? Because you have sequences already in place. Then there are 2 excision repair pathways that can also fix single strand mistakes. The first one is base excision repair, and the second one is nucleotide excision repair. Base excision repair is when you have mutations due to deaminated bases, right? So the example of that is where you have a deaminated C that's been changed into a U. It's only going to remove the base without removing the backbone. That's the difference here. On the other hand, in a nucleotide excision repair, you remove about 29 nucleotides fragment, similar to how we show the mismatch repair here, right? So it's going to remove an entire portion around the mismatch, around the problem. And this is typically kept for those thymine dimers, right? Or other pyrimidine dimers. Those are what's going to cause that. In that case, it just removes a whole portion of that backbone included before it fills it up. And so in this case, once in a base excision repair, once just the base pair is removed, the backbone is intact. So this other protein called AP endonuclease will remove the backbone just from that base pair and then refill it up. DNA polymerase will fill it up with a new nucleotide along with the ligase fixing the next. So it will usually correct the damage very simply with just taking that one single base out. In nucleotide excision repair, the whole section is going to be corrected, and it usually is meant for correcting bulky lesions that are distorting the DNA because of this cross-linking event of some sort that has happened inside it. Okay. Okay, so we'll stop here. And then next week, we will start with double strand repair or and talk about how that happens and how many, what are the different ways that that can happen. And then continue with the rest of the stuff. We will also finish up next time with apoptosis as well. I will actually see if I can post a little video. This shouldn't be more than 15 minutes of this as well. So you can kind of get a head start on it, especially since much of this is going to talk about P53 and caspases that are important for you to understand for lab as well. So by giving you that lecture now, I will be able to get you that information so that you can make better judgment for those questions that are related to your like cell viability and protein analyses. Okay.
Okay, sounds good. Thank you so much, Professor.
You're welcome, guys. Good.
See you next week.
Thank you. Have a good
See
one.
you next week.
Bye.
Bye.